Okay, so hello everyone, and thank you for joining us. I am Subrata Sarkar. I am the coordinator of NANO Global Project, and we have Dr. Lilian Krug with us. She is the scientific coordinator of POGO. So those who are not aware about NANO, NANO is the NF POGO alumni network for the ocean. So the scholars who received trainings from different programs of POGO, that is the different training programs supported by POGO, are the members of NANO. And our mentors is also the member of NANO. So the main objective of NANO is to maximize the benefit that trainee received during their training to get maximum benefits in their research career. And NANO has different activities. For example, we have a research project, we have networking activities, we have outreach and citizen science activities. And this NANO DOC is one of the research project of NANO. So the full form of NANODOP is a global study of deoxygenation, ocean acidification, and primary productivity at selected sites. So the prime objective of this project is to collect the in situ data of essential oceanographic variable from different areas. At this moment, we have sampling sites from 18 different countries of Asia, Africa, and Latin America. And the specific objectives of these projects are support in situ and promote satellite remote sensing observation of essential oceanographic variables at participating coastal sites, conduct a local analysis and inter-sites comparative analysis, organize capacity development activities, promote outreach and citizen science-based activities at these sampling sites. So in this project, essential oceanographic variables means temperature, salinity, dissolved oxygen, chlorophyll, and pH. But other sampling sites also collect other variables like nutrients, plankton, and some other oceanographic variables. And this webinar series is also part of this project, and we consider it as outreach activity of this project. So in this webinar series, we actually invite a speaker to share his or her recent research activities with us so that it can enhance our capacity in ocean science. And this webinar is open to all. That means anyone can join in this webinar, and we announce about the webinar in our social media platform. So anyone can register for this webinar and then participate in this webinar. And today we have Professor Dato Dr. Aileen Tan Shau Hwai from Malaysia, and she will talk about effects of ocean acidification on oyster aquaculture. Professor Aileen is the director of Center for Marine and Coastal Studies at Universiti Sains Malaysia, a fellow of the Academy of Science Malaysia, and serves in various international roles, including as Vice Chair of UNESCO, IOC Western Pacific, and board member of POGO. Aileen is the first women president of UNITAS Malacologica from 2013 to 2016. In 2020, she received the TRSM for expertise in mariculture and marine ecology, biodiversity, and conservation, and is recognized as the best women scientist for guiding women in Johore Islands. Professor Tan is a pioneer in green aquaculture in Malaysia, focusing on mollusk culture to provide alternative livelihoods for coastal communities aligned with SDG 1. A strong advocate for translating knowledge into community benefits, she holds several Malaysia Book of Records for molluscan studies, emphasizing a commitment to a sustainable future. Thank you, Professor Aileen, for accepting our invitation. And before we go to talk, I have some few issues to announce. This webinar has two segments. In first segment, we will have 30-minute talks from our speaker, and then we will have 30 minutes for question and answer session. And this webinar is being recorded, and we will upload this webinar in our YouTube channel. So if you do not want to be recorded, please switch off your camera. And during question and answer session, you can ask questions to our speaker, and if you can also write your questions in the text box in the message box so that we can transfer your question to our speaker. So thank you very much, Professor Aileen. Over to you.
Yeah, thank you, Supatra. A very good evening, good morning, and good afternoon to all the audience here, wherever you are. I'm from Malaysia, so it's actually here 9 p.m. in the evening. So I'm very happy to be here. Let me share my screen because you have looked at my face already, so it's time to share my screen. Hold on. Okay. Right. Okay. So I guess you can see my screen, right?
Yeah, working
Okay,
perfectly.
good. Yeah, good. Thank you. So the topic that I would like to share is effects on ocean acidification on oyster aquaculture. Many of us, even under the POGO program or in many other programs, we do a lot of monitoring of the ocean in terms of the ocean variables. And ocean acidification is one of the monitoring that most marine scientists is doing. But the questions I would like to bring your attention to is, after monitoring, what's next? If we know that is, pH is reducing, so what does that means? How does that impact to our livelihood? How does that impact to the living organisms in the ocean? That is why I would like, scientists not only should, that's why there is a few areas that scientists should explore, not just having regular monitoring. Yes, it's good to have baseline data to see what has changes over the years. And also we should be looking at what has the changes, what can the changes in the ocean variables impact to the livelihood of the communities surrounding the ocean. Yeah, so that's why we brings to the title of effect of ocean acidification on oyster aquaculture. Yeah, and based on the reference, in year 2009, it stated that climate change has actually has implications for the fisheries and aquaculture. And one of the several of the issues are increased carbon dioxide and also ocean acidification. I have to mention here the study on ocean acidification has started way before the studies on microplastic pollution. And why is microplastic pollution or plastic pollution or marine debris pollution is gaining more attention than ocean acidification? This is because ocean acidification is something not visible to our naked eyes. We have, when there's changing in pH, when the pH become reduced, the general public do not sense it. And the changes to the general public is very minimal. But that minimal changes in the pH in the water actually impacts the lives of the ocean in terms of biophysical effects, implication to fisheries and aquaculture, and also what is the status of the current knowledge. So how do we now bring, set the limelight on the ocean acidification and make it important, because it is an important issue that is impacting our ocean and livelihood. Yeah. And if you look at this figure, it was the source from the National Geographic publication. You can see from here, if CO2 continues to rise unchecked, computer models show that acidification will deplete the carbonate ions in much of the ocean by year 2100. Turning the waters corrosive for many shell-building animals. Why again, I would like to bring attention, this, it sounds like, so what? So what if is CO2 continues to rise and pH continue to low reduce. The shellfish, if you look at the crabs, look at the coral reefs, look at the oysters, to you or to those consumers, they will look at it with the slight change in pH. It doesn't impact the shellfish because the shells are so much thicker. You need strong acidic condition to dissolve it. But do not forget the life cycle of all the shellfish. The very beginning of when the shell is formed, the shell is super thin, and the slight change in pH will actually impact or stop the development of the shells, just like what been shown down here. Like shellfish been exposed to lowered pH. You can see, after 45 days, it become thin. And what I would like to show, for those from this region, Southeast Asia region, why is it blank, white in color? Because there is no data. There's no data been collected, not or I would say limited data. That's why they cannot do projection. They cannot do a modeling. Yeah, cannot do models to project what would happen in year 2100. Yeah. And knowledge, there is knowledge gap. Definitely there's knowledge gap in the Southeast Asia in terms of aquaculture. Yeah, on shellfish is very much impacted by the change in the global climate. Yeah, but we cannot have preparation. We cannot what we say, get ourselves prepared, because there's lack of field-based experiment. There is prediction, there is a lab skill, but there is no field-based experiment. Because when we do experiment in the lab, sometimes we'll reach a stage that we need to test it out in the actual environment. So for ocean acidification in Southeast Asia, there is definitely lack of field-based experiments, and of course also lack of comparative experiments. This has been has been in agreement with Mary Sewell, which is quoted in year 2012, which is 12 years ago, and we are still not progressing much compared to the progress in the research on plastic pollution. So we are not just looking at ocean acidification or aquaculture. We are now actually losing our bivalves. In this talk, I will be focusing more on bivalve, especially on oysters, because the experiments conducted is on oysters. Yeah, and losing, when I say losing our bivalve, it's just a statement. But what is the important indication is we are losing our food security, especially when we are talking about sustainable aquaculture. Sustainable aquaculture is is focusing on aquaculture on the lower trophic level, which involve the shellfish, the invertebrates, and bivalves are invertebrates. Yeah, so if we do not pay attention in what climate change can do to our invertebrates or bivalve or shellfish, they will continue. It will also lead to losing our food security. And I would like to bring your attention, in the Southeast Asia, it's such a coincidence. We are very high in biodiversity. This area are also very vulnerable to climate change. Yeah, and we would like to bring your attention to synergistic stresses in the Southeast Asia. Yeah, ocean acidification, which is low pH, high temperature, and also low salinity. Yeah, in layman terms, besides the salinity, in layman terms, we consider our ocean getting hotter, getting more sour, and it's getting breathless. Yeah, so that links to another topic, which is deoxygenation. Yeah, but in this talk, in this research that I'm going to share with you, we are just looking at low salinity, high temperature, and low pH condition. So the question is, if any organism, any bivalves been exposed to all these stresses, multi-stresses, can they survive in the tropics? Yeah. So to study the effect, to answer that, So a study was conducted to study the effect of ocean acidification in the tropics, where 2 sites and 2 local commercial oyster species were chosen. Remember in the earlier slide, one of the stresses is salinity. So we are looking at the commercial species used in the region here is mainly the mangrove species. Yeah, the mangrove species. That is why it's subject to high fluctuation of salinity. If you look at this, the map, yeah, in the lower left-hand side, this is where the study site is, which is in Malaysia, Peninsular Malaysia. And zoom in is just right near the Straits of Malacca. This is the South China Sea, and this is the Indian Ocean. This area, the down part is the Indonesian area, and we are looking at the site A and site B. I will describe what is the site A and site B later on. So the 2 local species being used is the mangrove species, which is Magallana belcheri. The earlier genus name was considered as is called Chrysoistria. Yeah, but now they have actually revised the genus into Magallana. So Magallana belcheri, and locally is well known as white scar oyster. And also the other species, the second species is Magallana bilineata, which is used to be known as Chrysoistria irvideli. And this is known as the black scar oysters. Because why I'm introducing the white scar, why is it called white scar and black scar? Because the adductor muscle, where for Magallana belcheri, the adductor muscle scar is white in color, and for Magallana bilineata is black scar. Because when we deal with the community or the growers, they find it very difficult to memorize the scientific name. So it's easier to actually communicate with them by using easy name, yeah, common name, white scar oysters and black scar oysters, yeah. And the 2 sites, yeah, 2 sites that we have chosen, yeah, site A and site B. The description, these are the photographic view. These 2 sites are actually oyster culture site, yeah, oyster culture site. Both is on the Straits of Malacca. So, the site A is about 1 kilometer from coastline, which is near an island. So it has a pH of 8.3, which is near the ambient pH of the seawater. And the mean salinity, because it's located near an island, is a bit offshore. So the mean salinity is around 30.3 parts per thousand. And the salinity range, yeah, the range is about 28 to 32 parts per thousand. And site B is along a river, Merbok River, yeah, subject to huge water runoff during rainy days. Yeah, so the pH in this site B is 7.29, which relatively lower than site A. And the mean salinity for this site B is 20.5 parts per thousand. And the salinity range, it can be as low as 7 all the way up to 25. Because when it's rainy day, it will actually brings down the salinity, yeah, of the area. Because I would like to show you the site, because site B is in the river, in along a river, whereas site A is offshore in the island. Yeah. What we did was the hatchery produced oyster seeds. That means in a very controlled environment, oyster seeds were placed of these 2 species were placed in site A and site B, meaning that both the species is placed in both the sites. Yeah? So the parameters measure for the first round of experiment, the parameters measure is the survival, the growth, and the feasibility to reproduce. Yeah. So let's look at the result for this simple experiment. Yeah. Always remember, site A is the marine site. Site B is the mangrove site. Yeah. Where you have lower pH and lower salinity. If you look at the survivor, the results of the survivor, let's look at site A. Site A, the survivor for the white scar, which is the black scar, which is Magellana bilineata, is 84.6%, whereas the Magellana belcheri is 75.8%. Compared to site B, which is the mangrove area, M. bilineata is 95%, M. belcheri is 82%. If you look at the figure, it looks relatively better survivor in site B. Yeah. And then but I would like to point out here, oyster at site B showed higher survivor. But statistically, there's no significant difference between the survivor of these two species between site A and site B. Yeah, it's just that in terms of number, but statistically, there's no difference. Yeah, but in terms of growth, yeah, it looks like that's a survivor we're mentioning. In terms of growth, both the species shows faster growth in site A, yeah, compared to site B. Yeah, so even though when you look at the survivor, both shares the similar survivor. It shows that both the species produced from the hatchery control environment, when exposed to different pH and different salinity, they are able to survive. They are able to tolerate acidic environment at this stage. Yeah, at this stage in the juvenile until adult stage. The shell thickness for site A is thick compared to site B. Yeah, in terms of reproduction fecundity, both species in both the site are still able to produce. Yeah, they're able to become matured and they're able to produce healthy larvae. Yeah. Okay, but then when we mention the shell thin or thick, yeah, for site A, this is just an example of what been observed in the shell thickness. For site A, where the pH is ambient pH with salinity of a marine salinity, you can see this is how it looks. Yeah, but if you were to look at the shell, yeah, the shell at site B for pH, where the pH is much lower, salinity is much lower, the shell is thin and it become very brittle. So when the shell is very brittle, if we leave the oysters longer in this site, it will be it's not the growth that is an issue, but the survival will be influenced by predators. Yeah, the survival would not is high if it's just the environment parameters. But then if once the shell has become thinner, it's very much prone to predation. Yeah, because it's thinner. So the crabs or even the drill snails will come and attack them much easier compared to site A. Yeah. So in terms of the production fecundity, yeah, the oysters from site A and site B, both the species were used as spawners. Yeah. So the larvae were produced and were cultured at different pH level. Yeah. So this is how the setup looked like. Running experiment to control pH is not easy. We have to have a stabilizer to regulate the pH. Yeah, in order to ensure that as we grow the larvae, the life cycle of larvae, it needs at least 2 weeks to reach settlement stage. So and we change the water every other day. So after changing, we have adjusted the pH. We have to make sure that the pH been regulated at that fixed pH under the experimental treatment. So that is the challenge that we have to face. Yeah. In terms of the results, yeah, for the larva survival. So larva survival, that means from day one after fertilizing them until pedivaliger stage. Pedivaliger stage is where pedi means the larvae with food. When the larvae has reached a stage where the food, yeah, this is the food visible, that means they are ready to settle. They are looking for place to attach. For oysters, once they have attached or cement themselves, they are stuck for life. That means they do not swim anymore. During the first 12 days, 2 weeks of their life cycle, they are swimming. So this is a result of the larval stage, the larval stage. Percentage of survival cut across the different pH, So the yellow bar is representing the larvae from broodstock from site A, and the bluish bar is broodstock from site B, You can see in general, you can see larvae from broodstock produced by broodstock from site B are more, they can survive at a bigger range of pH compared to broodstock larvae produced from by broodstock from site A, which is the marine station. Their adaptation ability is less, So look at here, broodstock from site A, larvae is unable to survive well in pH lower than 7.8. That's why you don't see the yellow bar going all the way to pH 7 or pH 7.2. Even 7.4 and 7.6, the survival is extremely low, compared to the larvae produced by broodstock from site B. And then broodstock from site B, the larvae were able to survive in pH as low as 7.2, although the percentage survival reduced from pH 7.6 to 7.2. So that's the tolerance limit of the larvae, depending on where the broodstock comes from. That means acclimatize, the broodstock acclimatized to marine species, marine water, then the larvae would not be able to tolerate lower pH. But if the broodstock are acclimatized to sites that is exposed to low pH, then the larvae are able to survive at lower pH. So you can see that there's actually no significant difference of the survival in the larvae produced by broodstock from site B. Okay, when it comes to larvae, the percentage of settlement, I have to mention here, for the black scar, Magallana bilineata, and for the settlement, we expose them to pH 25 ppt, because that is the optimum settlement salinity for the black scar. But for Magallana belcheri, we expose them to 18 parts per thousand for settlement, because that is the optimum condition. We want to prepare an optimum condition for the larvae to settle, because we want to look at specifically at the pH in this case. So this is the results showing percentage of settlement, how many of the larvae that is swimming settle if exposed to the different pH. Again, the yellow bar is representing larvae of Pedivaliger from site A, and the blue one is from site B, larvae produced by the broodstock from site B. And the stars indicating no settlement experiment was conducted at this pH level, because high mortality during the larval stage, we failed to culture the larvae until Pedivaliger stage to run the settlement, the percentage of settlement. So if you look here, look at the yellow bar. The yellow bar are only able to settle, successfully at pH 7.8 or and also 8, compared to the larvae that is from broodstock from site B, that can actually still able to settle all the way down to pH as low as 7.4. So this larval settlement of broodstock from site A was lower than site B at pH 7.8. So you can see these are some of the indication that we have highlight. And and larval settlement of broodstock from site B showed no significant differences between pH from 7.6 to 8. That means if the pH will go back to ambient temperature, they are still able to settle. But if the pH reduced to 7.6, they are still able to settle, because they have been acclimatized as broodstock during the broodstock stage at an area that is exposed to low pH. Yeah. So settlement could not be affected by different pH level if the optimum salinity is given during the settlement stage. Yeah. So the conclusion, what is the conclusion that we can draw from these two sets of experiment? So we can say in a general scope based on direct explanation from the results. Yeah, Magallana bilineata and Magallana belcheri are able to survive at sites with low pH level. I mean, as juvenile and also as adult. However, the growth and thickness of the shells are affected by the low pH level. Yeah, remember, when you're exposed to the low pH, the shell become thinner, and the survival would then be impacted by predators, not so much of environment conditions. Yeah, the larva tolerance towards lower pH level depend on the source, yeah, of the broodstock, where the broodstock comes from. If the broodstock comes from an area with low pH, the larva can tolerate lower pH. If the broodstock comes from marine environment, the larvae would not be able to tolerate lower pH. So this results, the question asked next is this an acclimatization responses, or is it an adaptation responses? If exposure of the broodstock to the environment of high pH or low pH for a short term, then the results will be explaining acclimatization. But if the broodstock is from the wild and we harvest it naturally from there, most probably a few generations, they are occurring then naturally, then it would be by adaptation. These answers, we would need to run the experiment. We have to run the experiment. Then we have to use higher biotech approaches. For example, going through proteomics analysis to look at the protein and how it links with the structure of the genetics in the animal itself. Yeah, but at this moment we leave it as is a adaptation, is a approaches using either acclimatization or adaptation. And then the next conclusion that we can make is the effect of pH would be insignificant if the optimum salinity is provided. If optimum salinity is provided during the larval settlement stage. That means if that means meaning that the effect during the treatment would not be significant if we provide the optimum conditions for the larvae to settle. Yeah. And I would like to bring your attention to the natural scenario, yeah, of this area. This consider, the earlier part is consider a refugia, a natural refugia to run ocean acidification experiment. So this, I put that inherent adaptation. That means it's inherent, yeah. Benthic organisms surviving in this current extreme area may display the positive carryover effect, because they have been there all the time. If you look at the Straits of Malacca, the one that dotted with red color, this area is subject to a huge runoff from rivers from both Malaysia as well as Sumatra. And it actually fluctuates and huge stresses of reduced pH. Deoxygenation is also happening in this area because there's so much of activities of pollutants going into the Straits of Malacca compared to the South China Sea. Yeah. So it is extremely, if you look at the color of the pH in this area, for the Straits of Malacca, this area is extremely marginal, and that is why we can say it's in line with what been proposed by Pauline Rose in year 2012. During that time is the possibility of the positive carryover effect. So species from this area, yeah, not just having the results. Shouldn't we be thinking about food security? If this species in this area is able to survive despite the reduced pH, shouldn't be species from this area be a good target species for aquaculture in the Southeast Asia, because they are already having this positive carryover effect. Genetically, they are tolerant towards low pH. Yeah. So again, bring your attention to increased CO2 and ocean acidification, the biophysical effects, how it impact. A lot of time we talk only about, we have this general statement, reduce pH, they will actually impact the shellfish and whatnot. But if we were to deep dive into the responses, yeah, it's actually, telling us it's a complicated responses depending on where are they acclimatizing until the broodstock, where is it from, depending on species, depending on the populations. Are the oysters or the shellfish originally from the area that is already low in pH, and also the responses of shellfish responds differently at different life cycle and whatnot. So when we talk about studying the biophysical effects, yeah, or the biological behavior responses of animals towards ocean acidification, it gives very complicated responses, and it's not like monitoring of the ocean using the probe. If there's no pollution, the pH is as it is, you just take it as a reading, but when you're dealing with living organisms, the responses is different, yeah, based on many factors. That's why I've put there complicated responses, yeah, on that. So with that, I hope that you do follow my talk, and now it is open for Q&A. Back to you, Subhatra. I will stop sharing. Thank you.
Yeah, thank you, Professor Aileen, for your very informative talk. And so now we will have question and answer session. And so if any audience, if you have any question, you can raise your hand, or you can just write your question in the chat box. So it's time for asking question to Professor Aileen.
If this is my class, if no question, I will ask question,
Maybe I can start with one question.
Sure,
Professor
sure.
Aileen, yeah, thank you for
Yes.
your talk. So I have actually two questions. So did you consider the environmental condition when you collected the brood? And then what type of culture materials did you use to settle this part? And did you check if the culture materials has any impact on the growth of the species?
Yes. For the brood stock, yeah, that we collected is we have two sites, site A and site B. So we actually collected a collective brood stock because these two species can be found readily in both the sites. Yeah. So when we collected black scar and white scar from site A, then we'll run the experiment exposing them to site A condition, site B condition. And then if we also collect rootstock from site B, and then bring it back to the lab and expose the roots, that means the offsprings, to condition of site A and site B. So we would know, the carryover effects from the broodstock, or is it just the larvae itself that could not tolerate such condition? And your second question on the couch, when we run the experiment in the lab, we usually will use crushed shell, naturally from the oyster itself, crushed shells, and we sterile the crushed shell so it does not have any other trigger or biofilm that actually induce the settlement. So we actually have that in control. Yeah.
Thank you. Thank you.
Thank you.
So, any other questions?
Or am I too fast that you could not catch?
Oh,
Because
I have one.
yes.
I was trying to find here the raise hand. Oh, there it is. That's me.
Okay, yeah.
Okay, while
Yes,
people
yes.
are getting their questions ready, thank you so much, Aileen, for the great talk and such an important topic. I'm not a biologist, and I was trying to follow, comparing the two species performance, let's say. And I don't know if I got it right, but the white oyster, the one that it's more adapted to the coast, um, that species showed thickest shell and fastest growth, but at the same time is the one that has worse chances of survival, right? And I was trying to, do you know the reason for that?
Because the faster growth, because that things that when we run the experiment out in the field, the thing that we could not control, the variable factors that we could not control is the food. Is the food. Phytoplankton are much more in the river system compared to the marine ecosystem, the marine environment. So that is why most probably the growth is faster in the mangrove area, and also the shell thickness is due to not only so much of it links, I have to let me construct the answer carefully first.
Of course, probably availability of material.
Yes, in the control environment, salinity, for example, the black scar and the white scar, the black scar would grow better in lowered salinity, in a controlled environment, the optimum condition. Whereas the iridally, the black scar would grow better in higher salinity. But when the broodstock have acclimatized themselves in different environment, so the offsprings would not show such preference, because of the conditioning of the broodstock. So that is why when we run the experiment of swapping the broodstock to different environment, the larvae from the broodstock from the ideal environment to the stress, the new environment, and vice versa, then we can actually look at how the animal itself deals with stresses in a new environment. It might be a bit complicating, but that's why because when we do physiological study, we have to actually grow the animal in the ideal condition and also grow the animal in the exposed treatments. That is why, but when we are doing it out in the natural environment, is more tricky compared to having a control setup in the lab.
Yeah.
I'm sorry if I'm confusing you more.
I think I need the chart.
Yeah. Yes, Sophie. Yeah, how are you, Sophie?
Hi, Eileen. Hi, everyone.
Hi.
Sorry, I accidentally had my video on at the during half of your talk, and I was preparing my lunch. So apologies if you
no worries.
anyone
I don't notice that.
And I also arrived a little bit late, so I don't know if I missed this. But my question is, it's not so much about the science, but about the aquaculture industry in Malaysia, and how interested are they in the topic of ocean acidification and in your work? Are they interested, for example, in providing funding for any of the experiments that you do? Are they interested in the results? And are they keen to monitor ocean acidification in their aquaculture pens or whatever you call them? Because I know, for example, in Latin America, in Mexico and some countries like that, there's quite strong engagement between aquaculturists and scientists to sort of work together on monitoring.
Yeah. Thank you, Sophie, for your question. I think yes. At least I can say on behalf for Malaysian side, we do work closely with the local authorities and also with the farmers, the coastal aquaculturists, because they know very well that whatever changes in the environment will impact their harvest, their production. And by knowing what is there, and by letting us work together with them to run some experiment, they would now know, because following to all these experiment, the scientists would advise them, changing of target species to culture, or harvest or using broodstock from different sources to culture in their area, because with the scientific findings, we can actually advise them on what are the hardier species, or the species with thicker shells, or species that can tolerate wider range of salinity, temperature, pH, or whatnot. So they are very keen and to listen. And of course, oyster farming now depends very much on hatchery seed. So we are also liaising with the seed production industry on what broodstock to use, or broodstock, the sources of broodstock to be used for their seed production and to produce juvenile to be distributed to the different aquaculture sites. Yeah, so we do get fundings, small funding, but not much, to run some experiment to actually get more results, to create an alternative options, because the behavior or the physiological behavior of the animal, sometimes it's not fixed, it's changing all the time. That's why it's not as straightforward as putting the probe and measuring the pH or DO from the water. When you're dealing with biological component, is a different ball game.
Yeah. Okay, great. Thank you.
Thank you, Sophie.
So thank you, Sophie. And we still have time to take few more questions. So if anyone want to ask any question, please, you can ask. Okay.
Hi, everyone.
Yeah, Chaima
Yeah.
Jamal. Yes.
Yeah, hello.
Hello.
So thank you for this interesting presentation. And I have some questions about the monitoring of ocean acidification. So I want to know if you made a permanent sea surveys programs, and how you do the ocean acidification analysis, such as pH, alkalinity, and how you do the calculation for the other parameters, if you do that. Thank you.
Okay, thank you, Chaima, for your question. We do, if you were to ask specifically the methodology, I would say I would not know, because we have a chemistry team who is working alongside with the biologists like myself on the pH monitoring, looking at the the content of the what contribute to the pH fluctuation in the area. But we also work closely with NOAA on deploying their arms into some selected sites. One of the sites are in the coral reef, and we actually retrieve it back after 3 years and look at what is there over the years. And also we have the arms and the cows and the bows. I'm not sure you are familiar with that or not. If you go into the website of NOAA, they have a section on OA, and they have a standard method that been used by all stakeholders or partners that they can actually contribute to filling up the gaps of OA issues globally. Shaima?
Thank you for your answer.
Yeah, if you can email me, I can link you with our chemistry team who is working on the chemistry component of the OA.
Okay, thank you. Because I'm a PhD student and I work about ocean acidification in the Northwest Africa. For that, I'm so interested to know how you do your analysis. Thank
Okay,
you.
I will type my email in the chat box. You can just email me and I will link you with the team. All right.
Okay, thank you so much.
You're most welcome.
Okay, thank you. So any other questions? Okay, so if we do not have any other questions, then we will close our webinar today. And thank you again for joining us. And thank you, Professor Aileen, for your informative talk. And we hope to see you again in our next webinar. Thank you.
Yeah, thank you. I've shared my email in the chat box. For those who are shy to ask any question, you can text me, you can email me anytime if you want to know more. All right, thank you very much. Thank you, Subrata. Thank you, Leka and Pogo, for inviting me over to share some of my work. Thank you. Good night.
Thank you,
and have a
Aileen.
good day. You're most
And you too.
welcome.
Enjoy the weekend. Goodbye,
Yes, thank
everybody.
you.
Thanks for your time.
Thank you.